In this unit, we've been discussing the development of the nervous system from the ectoderm. And in this lecture, we're going to focus on the most complex organ of the body, which is the brain. So we'll be looking at brain morphogenesis. And as you'll see later in this chapter, the brain grows from the inside out. But there are important changes that need to happen in order for the morphogenesis of the brain to take place. Let's begin by examining the different cell types of the central nervous system. And we're actually going to start by looking at the neural stem cells of the early embryo, and then we'll see how that population of neural stem cells is able to give rise to the different cell types of the CNS. We'll then review the morphology and the functions of the different cell types of the CNS, and we'll briefly examine some of the cell types of the peripheral nervous system. All of the neurons and the glia of the central nervous system are derived from neuroepithelial cells. And these are the cells that you're looking at in this picture. I know this looks like multiple layers of cells, but this is actually a single cell layer epithelium. Now, there appear to be multiple layers of cells because the soma or cell bodies are located at different heights along the apical basal axis. And you can see from this picture that the cells are polarized. That simply means that they have a distinct apical and basal end. At the basal end, which is the outer surface of the neural tube, the cells terminate in end feet. The apical end faces the inner surface of the neural tube. Now, these neuroepithelial cells are important because they are the first neural stem cells of the embryo. That means that since they are the first neural stem cells of the embryo, they're only going to be present early in development. So these cells are only found in the early embryo. And they'll give rise to two populations of cells. One of these populations of cells we've already encountered, and these are the ependymal cells. The other population of cells derived from neuroepithelia are the radioglial cells. The ependymal cells are supporting cells. I don't know if you recall from chapter 5 on stem cells, but the ependymal cells are eventually found at the neural stem cell niche. They secrete cerebrospinal fluid, and they're important for the maintenance of the neural stem cells. These are the cells that are eventually present at the rosettes or pinwheels of the neural stem cell niche, and recall that the association of the ependymal cells with the neural stem cells is important for maintaining the neural stem cell population. The other cell type derived from neuroepithelial cells are the radioglial cells, and these are simply the neural stem cells of the embryo, which give rise to neurons and glia. Now, in addition to serving as neural stem cells, the radioglia also serve as a scaffold for the migration of progenitors and newborn neurons, and these cells migrate to different layers of the brain. And we'll see why this migration is important later in the chapter. Before we examine the development of the brain, let's look more closely at the different cell types of the nervous system. And one of the major cell types is the neuron, which is depicted here. As you can see, the cell is polarized. One end of the cell looks different from the other end. As you may know, neurons conduct and transduce or change electrical signals into signals that regulate the different processes that occur in complex organisms. Neurons receive signals through the dendrites, which are numerous projections that extend from the soma or cell body, and these actually expand a lot during the first few years of life. Now, whereas you have many dendrites in a neuron, there's only one axon, which is a cellular appendage through which the signal travels to the next cell. The axon terminates in an axon terminal, and that's not shown in this image, which depicts a growth cone. And the growth cone is a projection that is present during development to sense the environment and to direct the migration of axons to their final destinations in the developing embryo. But as you can see, the axon is a single appendage. And it can be very long because in some cases, signals need to travel very long distances. For example, a signal generated at the spinal cord may need to travel all the way down to your foot. Now, the synapse of the axon is the location where neurotransmitters are released. Different cell types will be specialized for synthesizing different neurotransmitters. So neurons undergo a molecular differentiation. Neurotransmitters are released into the synaptic cleft, which is that space located between the neuron that is releasing the neurotransmitter and another cell. and the other cell can either be another neuron or another cell type. For example, a synapse between a neuron and a muscle cell is known as a neuromuscular junction. Neurotransmitters transmit the signal that generates at the presynaptic neuron to the postsynaptic cell, and this is the dendrite of the postsynaptic neuron. Once an action potential reaches an axon terminal, an influx of calcium triggers the exocytosis of neurotransmitters, which then bind to receptors at the postsynaptic cell. The other cell type of the central nervous system is the glia. There are three major types which we'll look at in a little more detail. These are the oligodendrocytes, the astroglia, and the microglia. Oligodendrocytes are the cells that insulate axons in the central nervous system, and they do this by producing a specialized cell membrane that's known as the myelin sheath. As you can see in this figure, the myelin sheath basically wraps itself around the axon, and that helps to prevent the dispersal of electrical signals. There are spaces between the myelin sheaths which are known as nodes of Rambier, and these are the only places along the axon where action potentials or electrical potentials can be generated. The result of that is that action potentials jump from node to node, and that makes the conduction of the signal faster. In the peripheral nervous system, there is a similar cell type, the Schwann cell, which is derived from neural crest cells. And we'll cover neural crest cells in more detail during an upcoming lecture. But just like the oligodendrocytes, the Schwann cells insulate the axons of neurons that are located in the peripheral nervous system. Myelination is essential for the conduction of electrical impulses in mammals, and in fact, the loss of myelination, as it occurs in conditions like multiple sclerosis, leads to a decrease in the electrical resistance of the axonal membrane, and this results in a loss of velocity in the conduction of the signal. Myelination is also essential for the survival of axons. The second type of glial cells are the astroglia, and these are a diverse group of glial cells that include the radioglia, which are the neural stem cells. Astroglia also include other types of astrocytes, which are important for things like the formation of the blood-brain barrier, responding to inflammation in the central nervous system, and supporting the homeostasis of synapses. Finally, we have the microglia, which were given their name because they're considerably smaller than other cell types of the central nervous system. And microglia play different functions, but they're basically like the immune cells of the brain. So they engulf damaged or dying cells in the CNS. And they exhibit behaviors that are similar to those of macrophages. And macrophages are cells of the immune system that also engulf dead or defective cells. And in fact, the microglia are not derived from the neuroepithelium at all. They actually arise from macrophage progenitors that are derived from the yolk sac and enter the central nervous system before the formation of the blood-brain barrier.